This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 70, recorded on December 12th, 2013. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, howdy. How you doing?
Today's 12/12/13, and yesterday was 12/12/12. No, it's not right.
No, it's 12,
That's last
11,
year.
13.
When is it going to be?
12, 11, 13.
Yeah, you're right. I'm all messed up. I thought I had missed a good day, but 12/12/13 isn't bad. And there is no 13/13/13, unfortunately.
But next year we'll have 12, 13, 14.
Yeah, that's good.
That's it. Okay, that's pretty good.
I like that one.
Are we going to get off that? Or
Yeah, we'll get off it. Also joining us today from Medical University of South Carolina, Michael Schmidt.
Hello, everyone. Great to be back with you.
It's been a few weeks, hasn't it?
Yeah, I was in Germany presenting our copper story at Medica, which is a large equipment show for, I guess, the world. I mean, imagine the general meeting of the society where you have all the exhibitors. Well, imagine 18 buildings of exhibits, all selling different types of medical equipment. And the physicians of Europe come. And so I gave a talk reflecting on our results from that May Itchy paper that we discussed in a previous TWiM. And I just learned today that in the EPIC guidelines for England, that we were listed as one of the emerging technologies for infection control. So we made it into a standard of care.
Nice.
So that's a rather interesting translational piece of science going from the bench in 2007 to a standard of care in 2013. So
Cool.
I don't normally sell my research, but I'm proud of all the people that have collaborated with us, and I think it's really why we go to work every day, is to facilitate a transformation in normal people's lives.
I think I was in Germany around when you were. I was there a couple of weeks ago, too.
Yeah.
you were at the giant virus meeting.
I was at the
right?
giant virus meeting, outside of Munich. Very cool meeting.
Well, you guys have all the fun. What can I
Yeah,
say?
we have a lot of fun. Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello, and it is 12/12/12 here in Ann Arbor, 12 degrees Celsius.
Wow.
I'm sorry, Fahrenheit.
You're kidding.
It's 12 Fahrenheit. Yeah.
That's cold, huh?
It's cold.
It is cold. Here it is it's below freezing. Yeah, I guess it is out there too. We can't rival that, but it's something like -5 Celsius. It's in the 20s here. And -3 now. How's your handicap? Good?
It's on hold.
It's on hold. You don't do any golf for a while, right?
Not much.
Unless you travel.
That's right.
Well, her graphite shafts will crack.
If it's that cold, I don't think she's going to be out there.
I'm cycling. I did cycle to work, I'm proud to say.
You cycled today?
Yep.
Did you wear a hat?
Yeah, and a helmet, of course.
And gloves, yeah.
Scarf,
Wow.
the whole bit.
Yeah, well, you chose to live there, right?
It's a great place to live.
I drive my kids to school in the morning, and there's this house on the route that has this Michigan flag hanging outside
All right.
all year round. And I think of my Michigan colleagues every time
I'm
I
glad
see
to
that.
hear that.
Why would someone do that?
Why
Because the
wouldn't
loyalty to our fine institution.
I guess they're probably, they really had a good time there. I do not hang a Cornell flag outside my house. I have to tell you that. Even though I had a fine education there, there's no need to. But then, it's all about enthusiasm for your alma mater, right? All right, we have 2 papers today. We are not going to talk about Michigan the whole time. I have 2 papers today. And the first one, Elio will present to us.
All righty. is called Bacterial Cytological Profiling rapidly identifies the cellular pathways targeted by antibacterial molecules, Symphoneus, and the authors are Nanuaji, Burkhardt, and the Polianos, Kit Poliano and Joe Poliano, who are both, well, they're married and they're professors at UC San Diego, and I know them well. So it's my pleasure to present this paper because these people are next door to me, and I love them and I love their work. So what they've done, to summarize what this is about, is a way of determining the mode of action of antibiotics in a quick and cheap way that lends itself to high throughput. Okay, what they do is they look at the bacteria. Simple as that. Now, here's the story. First of all, well, I don't quite know where to start because there's so much to talk about. But I'll try. First of all, when antibiotics act on bacteria, a standard bacterium, let's take E. coli, they change the morphology greatly. This has been known for a long time. Obviously, if you treat bacteria with small amounts of a beta-lactam, like ampicillin, not enough to kill them, they will stop dividing because cell division is more sensitive than growth. And they will continue to make filaments, and they will grow as filaments, in other words. And the nucleoids will be spaced evenly along the filament, just like they would be in a normal cell. And that's something you can see, easy enough to see. So I'll go over this, but the same argument goes for antibiotics that inhibit protein synthesis, antibiotics that inhibit RNA and DNA and so forth. So for the major classes, of which they say there are 5 of antibiotics assorted by their mode of action, there is a distinct morphology for each. Okay? Well, this has been known for a long time, I guess, but nobody's put it together. So what they put together is the thought that this is useful and allows you to sort unknown antibiotics just by looking at what they do to the bacteria after 2 hours incubation at an MIC or something like 5 times the minimum inhibitory concentration. In other words, the cells are barely growing, or they're really not, but they're not being clobbered by huge amounts of the antibiotic. Anyhow, what they do is they stain the cell membrane, and this is a cytoplasmic membrane, with a dye called, for those who care, it's called FM4-64, which is a red dye that stains only the membrane. And with DAPI, I don't know what DAPI stands for, and I don't care.
Chromogenic dye.
It stains DNA. So, okay, so they have...
diamidinophenylindole.
Good for you.
No, it's good for Google.
Okay. Anyhow, when you stain bacteria with these 2 dyes, you have a red membrane and a blue nucleoid. Now, I should tell you, however, that this is not that simple. What makes it not so simple is that to see these distinctions, one takes very powerful microscopy. And the lab of the Polianos is known for the exquisite use of microscopy, of optical microscopy. They have perfected it to a science, and they use not only the best microscopes available and all the techniques that are currently available for increasing resolution, but they also use computers in a smart way. They do a thing called the composition, is it? The composition?
The convolution.
The convolution, I'm sorry, of course. And the convolution gives them an increase in resolution just by itself. In addition, they measure under the microscope about 13 parameters, and the 13 parameters have to do with the width of the nucleoid, the width of the membrane, the thickness of the layers, all kinds of things, which give them, as I say, 13 parameters, which they can then ask the computer to organize by something that I hope the listeners, many listeners here know, namely principal component analysis. Okay. I know that Michael is an expert on that, and someday he ought to give me a lesson because I'm not sure I get it. But it is, if it's a way of actually. Michael, would you want to say something about principal component analysis, or am I springing that on you?
The simple metaphor to look at is most folks are familiar with Venn diagrams, where you ask where things interact with one another. You're looking for interaction zones. And I think if you look at principal component analysis, where you're looking at complex variables and you're asking specific questions and you're looking for trends or a schema, you put a lot of data into a bucket, I think, and you try to figure it out. The way I've always looked at it, in two dimensions, it's like a Venn diagram where you say, okay, these things are in this bucket, these things are in this other bucket. How many of them are coincident one another? Michele, am I getting it sort of right?
I think you're perfect.
It's all based on statistics and inference and probabilistic math of where things fall into place, but it's really a visual modality to condense a lot of data into one. And I think that's the gem in this paper is, the 5 targets of antibiotics were traditionally cell wall, nucleic acid biosynthesis, DNA or RNA, protein synthesis, cell wall division, and then, of course, specific inhibitors of metabolism or anti-metabolites. And I think if you look just at their first figure, and Elio may get to this in a second, but what really sold me on this was the visual images on that one first figure where they show the drugs and they show untreated cells, and you see bacteria cheat, and they have more than one chromosome, and you can literally count them with your finger on the untreated. And then tetracycline, which we all know stops protein synthesis. You see that you get all the nucleic acid content is condensed into one space. And rifampin,
moreover, the space looks like a donut.
Yeah.
Torus. And it is a torus. That's been known for some time, and nobody's ever figured out why they do that. But they do it.
And it's because the cell isn't needing to interrogate the DNA to make protein because protein stopped. And
that doesn't tell you why
it's a donut
shape, but it tells you that something serious is happening.
And then you just go through the figures and you look at rifampicin, which is against RNA, and you look at triclosan, which is, the evil. If you talk to Stuart Levy, it's the evil. And then you look at Cipro, which is DNA gyrase. And then ampicillin that causes cells to explode and causes them to elongate. It's so visually compelling. And I think, I always look at, neat ways to teach antibiotics to med students. And I'm going to actually steal this figure and show them to keep this in their mind to understand what's actually going on.
Terrific.
When you throw an antibiotic at a microbe, and it really teaches us the importance of how we use these massive, powerful chemicals, and we give them to people, and this is what happens to our cells. So I think this paper is going to go down as one of the seminal papers, and I'll bet you Big Pharma is looking at ways I
can tell you, but I can tell you that they are already. They are certainly, and the reason for that, if I may expand, is that knowing the mode of action. So this tells you the mode of action of an unknown drug. And in fact, in the paper, they use an unknown drug, and it's called what? Let me go back to
something or other.
Spirohexanolide.
Thank you. And they found out that it has a certain mode of action. In this case, it resembles niacin, bacteriocin. Anyhow, the reason. So why is this important? Why would anybody want to find out the mode of action of an antibiotic? And I asked Joe Poliano this, and he gave me one hell of an answer. Namely, the FDA requires knowledge of the mode of action for any drug.
Oh, yeah.
So it's something that is required by law. And in addition, of course, knowing the mode of action can help you in modifying drugs, can help you in looking for drugs if you wish. There's all kinds of angles in the drug discovery process where this becomes of direct importance. So let's take that for granted. So this is knowing the mode of action is important, and Big Pharma, as Michael says, is going to go after it in a big way because this is far easier. The alternative to finding out the mode of action is the old-fashioned way, which is to add radioactive precursors to proteins and nucleic acids and peptidoglycan and see whether these are taken up or not taken up in the presence of the drug. That's a lot of work. It's expensive. The other alternative is to use mutants in certain genes which sensitize bacteria to certain classes of antibiotics. That's also work. All of that is work. The beauty of this approach here, the beauty of this paper is not only does this work, not only can one distinguish various classes of antibiotics by the mode of action, but it can be done automatically. That is, they can take a slide with 96 spots, put it under the microscope, and ask the microscope to look at it, and the computer to decide what's going on by measuring those parameters I talked about earlier. In other words, this is almost no work at all. This can give you an answer within, well, not quite minutes, but you wait 2 hours for the drug to work, and then you put this under the microscope, and I bet you that within a day you get the answer to 96 different drugs. So this is pretty phenomenal. I think
or 384. Or 384. Right. The plates are getting bigger. My former graduate student who now works for a pharmaceutical company in San Diego, his companies, they have taken a drug from the bench to full FDA licensing, and it was based principally on a high throughput screen that used microscopy, very similar to what this paper is going to do for antibiotics. And this particular company's portfolios in G protein regulation. And what they did is they used a very sophisticated CCD camera to evaluate microtiter plates using frog melanophores, which change colors. And so this is a very cool variation because there are companies out there that have software that can analyze. So as you imagine taking a compound library of 20,000 compounds and having your indicator strains, you can pick MRSA, you can pick VRE, you can pick the nasty E. coli, you can grow them as biofilms or you can grow them as planktonic cells, and you can ask then the question, what's happening with these particular compounds, to simply answer a binary, do these compounds warrant further explanation? And this is probably our best hope for new ethical antibiotics.
By the way, the technique that they use, they call it bacterial cytological profiling, BCP, of course. And this is derived from, like Michael said, from studies in other systems. And you can use cytological profiling with animal cells, and this is where the term comes from. So we'll get to know this as bacterial cytological profiling, and it will be used very widely. There's no question. However, let me tell you that not everybody can do this.
Oh, no.
At the present time, you have to be equipped with very special microscopes and with very special computer power and Moreover, you have to have experience and brains and so forth. They point out, by the way, that as the more knowledge is acquired and the database is enhanced, that this will become more powerful. And in fact, they already can subdivide the antibiotics of a certain class into subclasses by looking at subtle differences. So the more data accrues, the more this will be useful. It'll be really a phenomenal way of doing it.
And you can imagine, too, that right now they're using 3 colors, but there are other colors available.
That's right.
There are plenty of tools that we have to label particular components inside the cell. So if they add just a 4th signal or a 5th signal, sensitivity will increase even more. So it's
other
fantastic.
Bacteria can be used. You can use gram-positives just as well. The lab, in fact, the Poliano lab is known for their work on sporulation in B. subtilis. So that's just obvious for them to extend this. But they use the E. coli here because they wanted to essentially come up with a proof of principle, which they did beautifully, and with the place where there are the most data, I mean, E. coli. So we got something new on our plate here in terms of technology and way of assessing how drugs affect bacteria. Like
it's very elegant, very elegant.
I was amazed that this works.
Yeah.
It's this paper is going to go down with 2 hybrid technology as being a way of evaluating what's going on in the cell.
Yeah.
I mean,
it depends how many people can do it. As Elio said, if it's not straightforward, 2 hybrid, is not so hard to do.
Yeah,
but
that's true.
So, Elio, what are these people going to train others to do it and seed the world?
I imagine they will. I mean, they have started a company of their own. Do this commercially, which is reasonable, and I wish them a lot of luck then they may not be so eager to train the whole world. But this is technology that's accessible. You can buy this in a way, and you can train people to do it. It's going to take them a long time, though. It takes a lot of experience to do this right.
I see.
But this is people who want to invest in it will invest in it. So anyhow, the main thing I want to say is how unbelievably simple an idea this is.
Yeah.
I used to play around with such things. I worked on what antibiotics do to the growth of bacteria, and I've known everything they say. And it never occurred to me to put it together and say, gosh, you could make this into a general technique for finding out mode of action of drugs. I never would have thought of it. I was always thinking forwards. This is thinking backwards, if you wish, in time. And it's just very, very beautiful.
That's really the key to science, isn't it? That someone puts it together. I mean, the ideas are out there, and I've had so many times where I've seen some result and said, I thought of those pieces, but I never put them together. But
that's right.
But when you have a bunch of people working, then someone's going to figure it out.
Yeah.
That's the beauty of science. Yeah, it's quite a nice paper. Very nice.
Yep.
All right. On to our second from Michele, which is all about pertussis.
That's right. The title, this is also a PNAS paper, recent. The title is Acellular Pertussis Vaccines Protect Against Disease, but Fail to Prevent Infection and Transmission in a Non-Human Primate Model. This is work from Jason Warfall, Lindsay Zimmerman, and Todd Merkel, and they work at the U.S. Food and Drug Administration in Bethesda. So let me start by describing the problem, and that is that whooping cough, as it's better known, is a worldwide problem. It's estimated that there are 16 million cases annually, and in particular, it's pediatric patients that are especially vulnerable. So we estimate there are about 200,000 children die each year worldwide from this disease, which is tragic because it's a preventable disease. We do have a vaccine that can protect from disease, and that's the focus of this paper. So historically, in the United States, there would be typically 200,000 cases of pertussis a year, and infants were dying. So there was need for a vaccine, and in the late 40s, a whole cellular vaccine was introduced and incorporated into a vaccine that probably most of us have heard of, the DTP vaccine. So this is a combination of diphtheria toxin, tetanus toxin, and then it was the whole bacterium, Pertussis, killed but formulated with the others. And that was just a fantastic success story in microbiology and in healthcare. The incidence, once we introduced that vaccine and it was used widely, dropped from 200,000 cases per year down to less than 10,000. So for 3 decades, the number of cases of whooping cough were actually quite low. And a couple things happened. A whole new generation of naive parents had no experience with this dreadful disease and perhaps got complacent. There were some concerns that the whole cell vaccine was causing some neurological problems, concerns that have never been established. And there was also
Excuse me, what do you mean by that? They weren't true, or they couldn't be established?
All the data says that it had nothing to do with the pertussis. Okay. And also there was variability from batch to batch. So for all these reasons, companies were interested in coming up with a more specific, more pure vaccine. So rather than use the whole bacterium, by then we understood more about the pathogenesis of this infection, and they designed a component vaccine. So isolated particular appendages from this pathogen and used those purified proteins in the vaccine instead of the whole bacterium.
And this is known as acellular, right?
Acellular. So it's a component vaccine, acellular. So that was introduced in the United States in 1991. So we're now 20-some years past that, and what we're seeing is an increase in disease. So 20,000, 30,000, 40,000 cases. That's right. So their outbreaks are being reported all over the country. Something like 49 of the 50 states had more disease in 2012 than we had in 2011. So there is much interest in understanding what's going on. And again, I want to emphasize that this is an awful disease, and I'm going to just play a audio of a small child who has this disease, and you'll see why it has the name whooping cough. So hopefully you could hear that child just gasping for breath and having these spasms, cough spasms. This coughing can persist for weeks, 3 weeks, 4 weeks, 5 weeks, and it is fatal for some small children. So the question is, why is the incidence of pertussis increasing across our country, and what can we do about it? So one of the strategies that Todd Merkel's group has taken is to take advantage of a baboon model to study transmission of Bordetella from one individual to another. And they have previously characterized this model and shown that baboons, like humans, get colonized heavily by Bordetella pertussis. And in particular, what's known is that this bacterium, its only known habitat is the human throat, the human upper respiratory tract. So it only spreads from one human to another. When we inhale it, the microbe has appendages that allow it to bind very tightly and specifically to the ciliated cells in our on our respiratory epithelium that are responsible for pushing the mucus up and out of our airways, keeping our lungs clear and keeping the airways clean of microbes and other particles. So this microbe can attach specifically to those cells, and then it replicates there and then elaborates or secretes 2 different toxins. One of them, called Tracheal Cytotoxin, actually specifically kills those cells that have the cilia on them. So those cells are shed from your throat, and you no longer have your mucociliary elevator, it's called, that keeps your the mucus moving up and keeping your airways clear. It has another toxin called Pertussis toxin, which specifically blocks recruitment of neutrophils to the site where the bacteria are replicating. And neutrophils are the white blood cells in our body that can move very quickly and they can eat very heartily. So they're really important at eating and killing microbes that are attached to our mucous membranes. So this microbe can attach, it can cause damage to the mucociliary elevator, which is a really important barrier, and it can also block neutrophil function. So the strategy then to design the acellular vaccine was to take the proteins that are required for binding to the ciliary cells and also to the toxin, the Pertussis toxin that blocks neutrophils from coming in and clearing the infection. So the way these experiments were done was to take populations or groups of baboons, and the baboons either had never been exposed to pertussis or they had been treated with the whole cell vaccine, the traditional one, or they were vaccinated with the purified component or acellular vaccine, the modern vaccine that's in use now, or the baboon had been previously infected and recovered, so had gone through the normal course of infection and now had an immune response. And the first question they asked was to now expose each of those populations of baboons to the live bacterium and ask, do they get sick and is there bacterial colonization in the airways? So in the first figure, what they show is that, in fact, the acellular and the whole cell vaccine are both very good at preventing disease, as measured in this case by increase in white blood cells in the bloodstream, or leukocytosis it's called. So both the acellular and the whole cell vaccine do very well, and in fact they do as well as a natural infection.
And Michele, that's because one of the hallmark indicators of infection is the amplification of this neutrophil population. So it's a very easy signal when you're developing vaccines to assess, because you just simply do a complete blood count or CBC and you ask the question, how many white blood cells are present and what fraction are neutrophils?
Right. And it's nice that in the baboon model, they see the same pathogenesis, the same host response. So they have a terrific assay for that. So that all looks good. The problem comes in when we now look at how successful are the pertussis at binding to the airways and replicating there. And what they show in their figure A is that if you've previously been exposed and recovered from the natural infection, you have no colonization. You're completely protected. If you've never been exposed to Bordetella, you get a very high level of colonization, more than 100,000 colony-forming units per mil of wash from the nasopharyngeal surfaces. Now, at 2 weeks' time, both the acellular and the whole cell vaccine both do pretty well. They knock that number down about 100 or 500-fold. However, if we look 2 weeks, 3 weeks, 4 weeks after infection, we can see that the whole cell vaccine treated animals or vaccinated animals are now have cleared the infection and they're probably doing quite well. But the animals that received just the components, purified components, still have really substantial levels of bacteria colonizing their airways for weeks before they eventually clear it. So that's the number one frightening observation, that the acellular vaccine, in fact, is not blocking colonization. It is blocking disease, but not colonization.
So, Michele, before you go on into the mechanism, let me throw out this question. The gold standard for determining a vaccine by the FDA is whether or not you make antibody. I mean, you use the antigen, you inoculate the animal or the human, and you ask the question, do they make antibody against your target? So based on this figure, should the FDA be considering a new standard vaccine metric from going from whole cell to acellular vaccines? in that we should, rather than asking the question, do you make antibody, should we be asking the question, does the new vaccine facilitate clearance? Because that, I think, is the gold standard in terms of what may be happening with the outbreak that we're seeing with pertussis, and why people who have been vaccinated end up with 100 days of cough afterwards, simply because the microbe hasn't been cleared from the system. So it's very similar to what the FDA does now for antimicrobials, in that what we require is an inferiority challenge. Is your new antibiotic better or worse than the existing standard of care? So in the case of a whole cell vaccine, which we know clearance occurs within, 14 days of challenge from that first figure, that should be the equivalent standard that the acellular vaccine should reach. Thoughts?
Yeah, you raise really interesting and important points. Let's come back to that after we talk about the last figure, because they do look in more detail at the immune response, and so we can then discuss what are better correlates of protection.
Okay.
It's a great point. So the next experiment, they simply ask whether animals that have been vaccinated with the acellular vaccine are blocked from colonization in a natural exposure. So they co-house animals that have either never been vaccinated or have the acellular vaccination, and find that both of them can acquire the organism, the pathogen, from a sick animal that is co-housed with them. The colony-forming units, the burden of disease is identical in those two cases. So again, if you've had the acellular vaccine, you can still, if you're coughed on by somebody who's carrying the organism, you can still be heavily colonized, even though you will not become ill. So then in the third experiment, they then ask whether animals that have the acellular vaccine and are colonized, whether they can spread their microbes to a naive animal. And that's shown in figure 3 in this paper. And as you would expect, even if a baboon is not ill, if they have heavy degrees of colonization in the respiratory tract, they in fact do spread it to naive animals. And because Bordetella is so easily transmitted through coughing and sneezing, anyone that's a close contact, a family member, somebody you might be next to on the subway or the airplane, will acquire colonization from your throat, whether or not you are sick. So they then want to look at this in a bit more detail and try to understand what the difference is between the host response for the whole cell vaccine versus the acellular vaccine. And before I go through that, I just want to back up and say that in general, when we're designing vaccines, we want the host response to be robust. We want it to be durable, to last a long time, so we don't need common boosters. But the host response also has to be the right shape, the right design. So to explain that further, you can imagine that if you're infected with a pathogen that is hiding inside a cell, the strategy for getting at that pathogen and clearing it is going to be quite different from the strategy that you would use if you just need to eliminate a toxin from the bloodstream. In that case, you'd want antibody. In this case, they have a microbe that is sitting on the surface of the respiratory epithelium, and they need neutrophils to come screaming in and eat those bacteria and kill them. To do that, we now appreciate that you need a special class of helper cells called T helper 17 cells, or Th17 type immunity. And
they've been discovered rather recently, haven't they?
That's right. That's right. And they make a cytokine, a signaling molecule called interleukin 17 that is known to recruit and activate neutrophils so they can come in and do their job. So it's possible to treat cells from animals that have been vaccinated either with the whole cell or the acellular, or have been through a natural infection, and then look at their array of immune cells and ask, does this have the profile of a Th17 response or a Th1, which would get at intracellular pathogens, or a Th2, which is more antibody protective response? And what they show in figure 4 then is that the whole cell vaccine, the traditional vaccine, stimulates to a greater degree these Th17 cells, which we now appreciate are important for activating neutrophils. In contrast, the acellular vaccine does not induce as much Th17 response, and instead it's a stronger Th2 profile, which we have no evidence would be effective against Bordetella. So to Michael's point, now that we can look in more detail in this animal model, we can see that the vaccine preparations actually trigger a different shape of an immune response, and then we can correlate that in earlier experiments to how effectively each of the vaccines protects. And we see that just overall antibody production is similar, in fact, after vaccination with the acellular or the whole cell, and that they show in figure 4. But the Th17 cells, which coordinate the immune response, respond differently.
So the Th2s, of course, are making cytokines that help mature the antibody response.
Right.
So that goes along with that skewed. It's not the right shape, as you would say.
That's right.
Yeah.
And it facilitates clearance.
Sorry, what facilitates clearance?
The antibody response.
Right.
If you also have the neutrophils.
Right.
Yeah, white blood cells.
So antibodies on their own will not
Right.
do it. You need to have the cells there, right?
Right.
So it's an antibody-mediated clearance, is that right? Along with the neutrophils?
And complement, probably.
Okay.
Yeah, so that they point out that one of the challenges with understanding pertussis pathogenesis is the mouse models are only good for studying certain aspects of the infection. The baboon model is clearly quite good, but it's
Quite
an
expensive
model. There are ethical issues with using a lot of baboons, and so the number of experiments that have been done to test the kinds of questions Michael was getting at, how effective a vaccine is in clearing infection, really requires this non-primate model or a human trial.
So we basically did not know before what the correlates of protection were. Is that a fair statement?
Well, immunology has advanced profoundly since 1991 when the acellular vaccine was licensed. And so that's one thing. You have to recognize that one of the outcomes of the last 15 years of the 20th century was HIV, and the immunologists learned a tremendous amount about vaccinology because of the effort that went into developing an HIV-based vaccine, which is effectively looking to prevent the infection. And as Michele so eloquently points out, we need to understand the shape of the correlates of immunity. And I think the pertussis situation really highlights the importance of understanding the shape.
So basically, Michael, what happened was that antibodies were correlated with protection, but we didn't understand that you also needed neutrophils or some cellular response along with it, right?
The cellular response,
yes.
the adaptive arm of the immune system.
So we really didn't fully understand the correlates of protection, which, by the way, we still don't know for HIV. We do not know what the correlates of protection are there.
It's a challenge.
Yeah.
So it's interesting that this acellular vaccine was first introduced in 1991. So our current population of children who are the most vulnerable have only had the acellular vaccine, which means they'll be protected. Each child will be protected from disease, but they will still be able to be colonized and act as a reservoir and spread that to other people. So most likely, we are seeing more disease in the U.S. because we have a higher burden of Bordetella circulating in the human population. And then if an infant who has not yet had their vaccine, has not yet developed protective immunity, is exposed, tragedy can result.
So that's a good argument for even more, until we have a different vaccine,
more herd immunity.
we should get more people immunized with the acellular to prevent
That's
disease, right?
Yeah,
and that's currently going on at our medical center. We're vaccinating the 20-somethings. We're boosting people right now because the 20-somethings are actually coming down with frank disease. They will actually get Bordetella. When you screen them for infectious agent, they're absent, but they end up with 100 days of cough. And if it's colonized and they're expelling these fomites, it really places, especially if you're a pediatric caregiver, it really places the vulnerable populations of pediatric patients at high risk. So it's part of our employee health plan that you can get boosted for pertussis if you're in that aspect of healthcare.
Actually, let me bring up another question here that I hadn't thought about. Being that this is purely a human disease, I think, unless you infect baboons and mice in the lab, why is this not a candidate for eradication, like smallpox and maybe polio?
I think it is, yeah.
Because it seems to me that if everybody got the acellular vaccine, the problem would be gone.
Well, don't go too quickly.
Not the acellular, because the acellular won't block colonization.
Right.
Yeah, but everybody would be immune against it.
There's a paper that's going to be published next month in Clinical and Vaccine Immunology that I just found this morning going through my table of contents emails. And what they have shown, this is from the CDC, from the first author, Lucia Pawlowski. And the CDC went back and screened 1,300 isolates dating from 1935 to 2012. And as Michele has just reviewed, vaccines, the acellular vaccines for whooping cough contain 3 to 5 of the protective antigens that are present for the vaccine's clinical effectiveness. But one of the antigens the CDC has learned, Pertactin, Pertactin, which had been present in almost all of the Bordetella pertussis strains circulating in the United States as late as 2010, is now absent in more than half of them. And so what this is showing us...
Half the strains, not half
the strains, that's right.
Yeah, half the circulating strains. So the acellular vaccine, because it does not prevent colonization, is actually selecting out an escapee that can escape the protective functions of the vaccine. You still have antibody because that's how we measure whether or not you've been vaccinated. But now, because of these outbreaks that have occurred in Washington State and in the Western United States, what we've seen, and the CDC went back and they screened 1,300 isolates, and using PCR, they found that there was an insertion sequence element popped into the Pertactin gene. And of the 1,300 isolates from 1935 to 2012, what these authors were able to demonstrate is that the first time they found the insertion was 1994. That's 3 years after we introduced the acellular vaccine. And of the 1,300 isolates, all after 1994, 267 of such isolates now contain some deletion, insertion, substitution in this Pertactin gene. So clearly, it's illustrating how the dangers of this acellular vaccine, we really need to get the shape of this curve right. And that's why I think this T-cell biology and understanding Th17 and the way I phrased the question to Michele about clearance, which takes into account how the immune system is working, may be a better readout for determining the stability of a vaccine in a population setting.
Which requires that you either do a human trial or you use you have an appropriate animal model.
Yeah.
And that's a challenge for many infections.
Oh, yeah.
Well, I mean, given the limitation of any animal model, right? That's why we call them models.
Right.
I don't think based just on these data that we would, make
Throw
a new
it out.
vaccine. I think you have to do some human studies, right? And now you know what to look for.
Yeah.
Yeah, and there are a number of clinical reports that are completely in line with this this study, showing that children who only had the acellular vaccine have a less robust immune response when challenged. Now they're more apt to have disease. So they've done retrospective epidemiological studies and found that even receiving one dose with the whole cell vaccine reduces your likelihood of becoming ill by like 8-fold. So there's pretty good data. I'll add just one more from a pathogenesis point of view. Really interesting. One of the components of the acellular vaccine is a hemagglutinin, a large adhesive molecule on the surface. Peggy Cotter's group has shown that molecule likely suppresses the Th17 response in an animal model.
Oh my goodness!
Wonderful.
Yeah, so it's like an independent point to Th17 arm of the immune system being really critical for pathogenesis.
So do you think that, is anyone considering going back to the whole vaccine, the whole cell vaccine?
I don't know enough about it to say, but if for my grandchildren, that's what I would be asking for.
I don't think you can get it in the United States. I don't think you can get the whole cell vaccine. I don't think it's... I don't think you can go to your pediatrician and say, may I please have the whole cell vaccine?
Yeah.
I just don't think they can
so
get
is
it.
That companies got out of that business because of the concern?
Well, it was reactogenic. The parents were uncomfortable with how the child behaved after vaccination. They were very uncomfortable. It was, it was highly immune stimulatory.
Which is probably great.
Yeah, it's why it works so well.
Pay me now or pay me later.
That's right.
Well,
that's right.
I think that's, one of the things I think we need to look at with acellular vaccines is the whole notion of adjuvants and what goes into an adjuvant. And in olden times, they had Freund's Complete, which is extremely reactive. And now there's Freund's Incomplete. But today, most of our vaccines have very little non-antigen dependent immune stimulators.
Right. And just to clarify, reactive, by that you mean some fever, some swelling, some pain,
discomfort.
at the site. Yeah.
Discomfort, anxiety, pain, anything that will keep your 2-year-old up all night to drive you to the Jack Daniels.
And speaking of 2-year-olds, our first author for this paper, Jason Warfall, he's a postdoc in Todd Merkel's group, and we had an email exchange. He actually he and his wife had twins join their family just 3 weeks after he started working on Pertussis. So I'm sure that as he was working, he was quite motivated to understand better how we can protect the pediatric population.
I mean, more importantly, the young adult population, because this disease is not pleasant for young adults because you'll end up with a cough for literally 100 days. You'll be sick. You'll have a frank bacterial infection type syndrome for about 2 weeks, and eventually your immune system will begin to clear it. But then all the collateral damage done by the toxins takes about 100 days to repair itself.
Yeah, you've got to rebuild all that ciliated micro epithelial cells. So
you got to rebuild a conveyor belt. And, I don't know about you, but conveyor belts are complicated.
So I have a short story I'd like to tell, which we actually did a related story many, many moons ago on TWiM. But I'll tell it again because, as Andre Gide said, there's no problem repeating yourself because no one listens anyway. I received the whole cell vaccine because I'm an old guy. And when I was 8 years old, my father took us to Italy to visit his family. And I slept in a room with a cousin, and she had whooping cough. And she coughed all night for like 2 weeks. And I'll never forget the sound, but I was in that room and I was fine because
you got a great natural booster.
Probably. And the thing is, my father was a physician, and you would have thought that, but I guess he figured I was immunized, and so he said, no problem. And if I wasn't, I would get immunized naturally. So I'll never forget that cough. I heard her cough. She must have been my age or a few years younger.
So
wow.
That whoop is when they're
trying
to grasp that next breath.
And I remember the first time I heard it, I got up in the middle of the night and everybody was still up. I said, what is that? Oh, that's whooping cough. That's the way it sounds.
I for the show notes for Ray, I pulled out a YouTube video that actually shows a 2-year-old in the full paroxysmal cough, and you can see how uncomfortable the child is because they're afraid they're not going to be able to breathe.
Yeah,
and the video really illustrates how uncomfortable the child is. And fortunately, she's got supportive parents telling her that everything is going to be all right. And they know how to calm her and to slow her breathing, because when you can't breathe, the intent is to take a deep breath that then, of course, sets it off even more. And it's really something to see if you've never heard whooping cough or actually seen an individual with it. And the young adults actually can get that same sort of breathing discomfort. And with the incidence of asthma in the population, it can be pretty significant, because there are many young adults that don't have their asthma all that well controlled.
Yeah.
And the vaccine is super important because we don't have a good treatment. So once you've got the infection, you just have to be, you treat the symptoms, but you can't cure
You're patient.
Yep.
Yeah, there's no magic bullet. Like, you don't write a script for a common antibiotic and you're fine in 48 hours.
But the reason is interesting. The reason is that it's not the bug that causes the disease eventually. It's the residuals from the bugs.
Oh, yeah. It's
so
nasty.
that's really a very difficult disease to treat, because you're trying to get not after the antibiotics, but the toxin that's there, and this toxin just sticks around forever. So
well, and it's caused all those
problems.
ciliated cells to leave, so you've got to regenerate.
Rebuild the conveyor belt.
This is a great story. Really, really nice story.
Lovely data also.
Beautiful. And I think it explains a lot, right?
Yeah.
I think
it's great. It's really nice. Thank you, Michele.
My pleasure.
I have a couple of emails I'd like to share. The first one is from Jim, who writes, just heard TWiM 67, excellent, of course, and recalled, like Elio, being in basic training in the 60s when meningitis cases occurred on the base, and we had to sleep with all the barracks windows open. I think we were double bunked, but I can't recall if we slept head to foot or if adjacent bunks were reversed. It was a cold time of year and open windows didn't improve the experience, but it seems like they were closed after a couple of weeks. I don't know, Elio, remember you were talking about sleeping head to foot?
Right.
Oh, yeah.
Yeah, in the barracks. That's exactly what we did.
So you struck a nerve with one of our listeners. Next one is from Richard, who writes, good morning, day, evening, depending on your time of day, esteemed professors. Firstly, my weather report for Weston-super-Mare, UK. It's currently 3 degrees centigrade, dew point 4 degrees, humidity 78%. There's been 1 millimeter of rain sleet with a 50% chance of further precipitation, wind 16 kilometers per hour. Predicted high is 9 degrees with humidity of 68%.
Chamber of Commerce weather.
I hope the weather report meets your increasingly exacting requirements. My question is fairly simple. Giardia lamblia and a number of other eukaryotes lack mitochondria. Most of them appear to be anaerobic, and I can see the point that the mitochondria and electron transport mechanism might be well selected against. However, nowhere can I find if it is clear that mitochondria were selected against and lost. Or if these bugs are a branch, then what started before eukaryotes adopted mitochondria.
Oh, boy.
I'm not a scientist, but simply an interested party. I actually work as an engineer on sewage treatment plants, so I do get to see a lot of bacteria since they do all the work treating the sewage. Given plenty of oxygen and the correct nutrients, they do a fine job of this and then happily settle out, leaving clean enough water that it can be returned to the environment or with minimal treatment and filtration to the drinking water supply, as is becoming more popular. However, it is my personal theory that apoptosis in eukaryotic cells derives from the incorporation of a once parasitic bacterium, such a parasite requiring a method to kill the host cell in order to proliferate into the medium to infect other cells. I hypothesize that this was co-opted by eukaryotes in order to allow for apoptosis or programmed cell death. Knowing if apoptosis occurs in eukaryotes that lack mitochondria and if they are a pre-mitochondria branch would answer my question. However, I've been unable to find the required information. I hope you can point me in the right direction. Many thanks for your ongoing series of podcasts.
Well, this guy has stumped me because this is something I didn't even remotely ever think about when it comes to Giardia. I mean, that is a really good question. And I don't know how old Giardia is and where it fits in the kingdom tree.
Well, the prevailing view, I think, these days, this has been argued a lot, still is. I think the prevailing view is that it lost its mitochondria, not that it was a branch that arose without them. And that makes, it's more appealing, I guess. Anyhow, that's about all I know. But I think the point that he makes is that this really is important for treatment and important for thinking about the disease. Sure is. But beyond it, I don't really know.
So he says, I don't know if it's true that Giardia don't undergo apoptosis. I seem to...
that's a good question.
I mean, that's really it.
Even bacteria undergo apoptosis.
Yeah. I mean, I'm not sure. I just don't know the literature. I wonder if anyone, maybe some of our listeners know, but it's a really interesting question. So any eukaryotes that lack mitochondria, can they still undergo apoptosis?
They found that bacteria have genes for something called metacaspases, which are related to the caspases which carry out apoptosis in eukaryotes, and that this may be the origin of caspases. In other words,
sure.
all of this came from bacteria, in which case Giardia would have it.
Right.
Well, we'd expect it to have it anyhow.
Right.
Yeah.
Next one is from John, who writes, Dear Twimmers, just saw this article about the rise of drug resistance, and he links to an article by Maren McKenna, who everyone should know as the author of Superbug, and it's called Imagining the Post-Antibiotics Future. It's a very nice story about someone in her family that died before penicillin of an infection and how it segues into the current problem with antibiotic resistance. He writes, it seemed a bit sensationalized, but as far as I could tell, was pretty good on the facts, and I thought it might make an interesting listener pick of the week. I'd love to hear what you all think of it. Reading the article, I wondered what we could do to keep resistance from arising in new antibiotics. For example, A, could a drug company refuse to license a new antibiotic for veterinary use or for agricultural use somehow? Ideally, we would just ban mass agricultural use of antibiotics, but who knows whether that will ever happen.
There's been good news on that front, actually, just yesterday.
What's that?
The U.S. government is changing the guidelines and making it more difficult for, saying that
today, this in the news today.
No longer include it in animal feed. No longer include antibiotics in animal feed.
Yeah, so this is slowly happening in Europe, and I think eventually we'll have some momentum.
Yeah, there was just a policy announcement yesterday, I think.
Yeah, I don't think that a company would refuse to license anything that it made.
No, if it's taken it that
far.
More
than half the market is to the agricultural.
Yeah, absolutely. B, could someone develop 2 new antibiotics with different mechanisms, but only sell them in a combined form? That would probably slow down development of resistance, sort of like the use of multiple antiretrovirals in treating HIV. That doesn't happen, right, Michael?
No, there's a lot of talk about going to doing that. In fact, many of the infectious disease docs, when you have a drug-resistant infection, actually use 2 and 3 antibiotics to try to effect a cure. But it's something that they're applying to the new hepatitis C regimen, they're using a 2-3 drug cocktail in order to move things forward. And, HIV, again, has taught us many valuable lessons. And because the virus grows so effectively in us and hides from our immune system, we had to resort to triple therapy at least in order to control viral load. And so if we could go back in time and we had the luxury of having antibiotics available to play that game, I think it may have been something that we would have started, and we may have had more antibiotics. But
so my view is that for every virus, you need a specific antiviral, right? So they can sell triple HIV antiviral therapy because it's just used for HIV, but for bacteria, you use some for multiple infections. So I don't know that you would want to sell a 2 or 3 antibiotic cocktail, right? You would let the physicians do the mixing.
Yeah, you let them do the mixing and matching, and it's based on the antibiogram that's coming out of the patient. What they're resistant to. So you don't, if you give an antibiotic that they're already resistant to, it's sort of like giving the placebo.
Yeah. And part C, would it be possible to develop a new antibiotic and then evolve resistant microbes and try to redesign the antibiotic to overcome the resistance? If you went through a couple of these cycles of changing the antibiotic and then evolving resistance, is it likely that would yield a final product that was hard to evolve resistance to, or would the microbes just find a different way to be resistant?
In fact, that's what Big Pharma did for many years, is they would take their candidates, they would screen them and ask the question under challenge how quickly they adapted.
Right.
And of course, the candidates that made it out of the Petri plate phase and got scaled into safety trials, of course, made it past those first few rounds. And it's in the dynamic equilibrium of a host that you really see the selection pressures impacting on the microbe and impacting on the utility of the antibiotic, how long before the organism adapts or dies.
But in the end, the microbe will find a way.
Oh, yeah. It's Perlman's law, the law of microbial infallibility. There's not been a naturally produced product on the planet that microbes can't overcome.
Right.
And for the most part, even the artificially manufactured products, microbes can overcome.
Right. Thanks for your wonderful podcast and for answering my amateur questions. They're great questions.
Terrific questions.
Not at all.
And, unfortunately, the pharmaceutical industry is interested in profits. And
yes.
Yeah, these John's ideas are really clever, but they ramp up the cost by billions.
Yeah, that's it. That's absolutely it.
I think our best hope for Big Pharma is going to be something like the PNAS paper from the Polianos, and coupled with thinking about probiotics to treat chronic diseases like type 2 diabetes, hypertension, and mental illness.
And vaccines to prevent the infection in the first place.
That's right.
Yeah.
Yep.
Ta-da.
We're still going to have them for sure.
No, we definitely need vaccines, and that's, I think, one of the most important things that we have is the indemnification of companies to encourage them to stay in the vaccine field.
Right.
Vincent can probably talk about, escape mutants of polio that still cause disease, even though you've been vaccinated.
Actually, they don't occur.
They don't occur?
No.
But I thought that's why we went away from the live vaccine.
Well, the vaccine itself reverts, right? It becomes neurovirulent, and that's a problem. But interestingly, the vaccine that was developed, 60 years ago still works today. Yeah. The virus has not undergone any significant antigenic variation. There are still 3 serotypes, and they are the same 3 as were 60 years ago, which is amazing. And, we don't know why. But the vaccine is effective. But the problem is the infectious vaccine, yes, it reverts. So that's another issue.
And that's why we went away from the live vaccine that gave us all that tremendous herd immunity.
Yes, in the U.S., we don't use Sabins any longer. We use the Salk vaccine, although you can still get it, I understand, because, in fact, now if you're traveling to Israel, the CDC recommends that you get the Sabin vaccine because there is poliovirus in the sewage there in Israel, and people are concerned about it.
I have a question for you. I got asked by one of my transplant surgeon friends whose daughter is going over her winter break to Madagascar, and he asked me because the travel clinic associated with her school was concerned that there's an outbreak of bubonic plague in Madagascar, and he was wondering how he should protect his 19-year-old daughter from Madagascar. And the only advice I could give him is tell her to sleep in clean rooms and watch out for fleas and rodent dung as an indicator of risk.
Yeah. Can she take some antibiotic with her?
Yeah. The drugs of choice are pretty spooky ones. The one that has the best efficacy in humans is an aminoglycoside, a variation of streptomycin, which has all its associated side effects. Gentamicin hasn't been used in people. And then, of course, going down the list, Cipro is supposed to have activity, but of course, it's never been properly studied. So, we're going to let the experts worry about that. I think he's talking to his friends at the CDC.
That's the place to go.
That's the place to go.
By the way, this article by Maren McKenna is very nice, and it's open access, and so I recommend you have a look. And that'll do it for TWiM 70. You can find this episode at iTunes and at microbeworld.org/TWiM as usual. And if you like what we do, go on over to iTunes and rate the show or leave a comment. That helps to keep us very visible there in the podcast directory. If you do have questions and comments, we love to get them. Send them to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
My pleasure.
And currently unable to work on her handicap because of the weather.
But looking forward to my Wolverines taking on the #1 team in the country here at Chrysler on Saturday.
Is that Alabama?
No, this is basketball.
Oh, football's over.
They're not discussing football.
Wow, good luck with that.
Thanks.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Well, my pleasure, of course.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, everyone. It's a fantastic show today.
Michael, you have nice weather down there?
Warm? It's not warm. Last Friday it was 80 degrees, and today it's seasonal. It's about 50 out.
Well, my wife is now a resident of North Carolina, not too far from you. She's working there 4 days a week.
Oh, my.
At her new job. So
Frequent flyer miles.
I think about you all the time. Yeah, she's flying back and forth. I'm Vincent Racaniello, and you can find me at virology.ws. Many thanks to ASM for their support of TWiM, and to Chris Kandayan and Ray Ortega for making it work behind the scenes. The music that you hear at the beginning and the end of TWiM is composed and performed by Ronald Jenkins. You could hear more of his stuff at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.